cells need quite a lot of looking after. They're quite needy, a bit like babies, I suppose. I'm Marianne, I'm 25 years old. After school I went to university in Nottingham to do a degree in biochemistry and genetics, where I decided that I'd quite like to go into cancer research. So I applied for a PhD between London and Cambridge, and now I'm firmly in London finishing my PhD. I have about a year left. So this is a Cancer Research Institute. Our group, we specialise in solid tumours, so that's any tumour that's not of the blood and the bone marrow, but any solid tumour growth. And specifically we look at the growth of blood vessels into those tumours, which is like the roots of plants that feed the plants. So for a tumour to grow, it needs blood vessels. So in our group we look at the cells that make up the blood vessels, why they grow into tumours, how that makes tumours get bigger, and how we can stop them from getting bigger by cutting off the blood vessel supply. So my PhD specifically, I'm looking at a particular molecule inside the cells of the blood vessels and how these cells stick to each other and move around. And can we use that in cancer research by perhaps designing drugs to block these? So this is a tissue culture hood, and it's a special, it's a bit like a giant fan, so it will suck air in a particular direction and blow it out of the top. So it's always got a directional airflow and it keeps things moving, so you don't get bacteria settling in your dishes. So we keep cells alive in the lab in tissue culture. We have to feed them, make sure they have the right kind of environment, make sure they have enough space and the right kind of ingredients in their food and that kind of stuff. So that does take a lot of our time, and they're quite picky. Here are my cell culture flasks. They're made of a special kind of plastic which has a coating on that cells really like to stick to. So this is our cell food. It's called growth medium. It's nice and red, and the red is because it gives us an indication of how acidic it is. So we can get our cells out of the incubator, have a look at how red or not the media is, and we'll know then that we need to change it and give them some food. So we have to warm it up first in the water bath, which is set to 37 degrees C, which is body temperature. Again, they're picky about acidity, they're picky about temperature, and a lot of other things. So when you're feeding your cells, the first thing you want to do is take off the old media, the old food that they've used all the nutrients from. So we use a vacuum pump for that. And all the waste you have from the cell culture needs to go into disinfectant to kill whatever's on there, because we don't want it getting outside. Even if it's not really that dangerous, it's just good practice to kill any bugs that you're not using anymore. So this is just it's a bit like water, but it's got some salts and things in it, so it's just a wash to wash off any of the old medium that's still in the flask. So this is it's a basic medium that we use in our group, specific for the kind of cells that we grow, which are cells that line the blood vessels. And then we just put the lids back on. So the lids are special. They have a kind of a filter, and they stop bacteria and stuff getting in, but they still allow circulation of gases. So again, the cells don't get too acidic. From the cells that we grow, we can take specific things out of the cells, like maybe proteins, maybe RNA, maybe DNA, and look at how these are changing in different situations. That maybe we're treating the cells with drugs or something like that, and we want to look at how they're responding on a more molecular level than the cellular level. So probably the worst thing about working with cells is that they have no notion of time. You can do your best to plan your experiment so that maybe you'll set it up on Monday because I wanted to finish it on Friday, but maybe for whatever reason they're not growing very quickly, and instead of being ready on Friday they'll be ready on Sunday. There's nothing you can do about that. You have to come and do it anyway at that time, but if you get the results at the end, then it's worth it.